My name is Dr. Paganotti Giacomo. I'm from University of Pennsylvania, and this laboratory is a joint facility between University of Pennsylvania and University of Botswana. And I have an appointment at the Faculty of Medicine, Department of Biomedical Sciences, as a senior lecturer. So this facility is able to analyze DNA, human DNA as pathogens DNA. In particular, we in this laboratory we study HIV-related topics. We study papillomavirus, and we study malaria, and recently breast cancer. So we have several projects. They are all funded from grants. We have internal UB grants as well as external from National Health Institute in USA, as well as Harvard University, etc. And we also are part of important networks. One is a sub-Saharan network for HIV and TB excellence in research called SANTE, based in Durban, that funds parts of this research. So in all of these different fields, malaria, HIV, papillomavirus, and breast cancer, we have students that they do their project on this topic. And for example, I can introduce you some of the postgraduate students running the project in this lab and being supervised from myself.
Yes, my name is Laban Ntawe. I'm a PhD student who recently submitted a thesis for examination, so I should be defending soon. So basically my work was based on studying the effects of HIV infection on the distribution of HPV genotypes and host epigenetic changes associated with cervical cancer progression in Botswana.
Okay, my name is Ginewen Tunto. I am a current new MPhil student at the University of Botswana. Well, I currently started my studies in March this year. So I am currently working on breast cancer pharmacogenetics. So this is where we have established that breast cancer is one of the common cancers occurring among women in Botswana, second after cervical cancer. So we have realized that there is a high mortality rate of breast cancer in Botswana, and in order to understand this mortality rate, we want to explore the pharmacogenetics behind breast cancer treatment. So what is pharmacogenetics? Pharmacogenetics, we understand that drugs do play different effects among different people. So in that case, they do differ in terms of toxicity levels as well as the way people respond to treatment. So those may vary in inter-individuals and at population levels at large. So our current study is mainly to understand the two main DNA variations that do alter treatment outcomes for individuals. So we do understand that DNA variations do influence drug concentrations and also how the times that drugs spend in an individual's body. So we realize that that is a notion that has been well studied in Botswana. So should that be true, the study is based on the basis that it will help classify individuals into poor metabolizers, normal as well as fast metabolizers. So with that, it will help clinicians in a sense that they will be able to alter drug dosages as well as concentrations for individual patients, which is known as personalized medicine.
So this, I want to stress in this context the importance of science, that is very important to understand the life around us, and it's very important at any level, from very young kids to men of my age, that science is important, because whatever we touch, whatever we do, whatever we listen in our mobile, or we play, or we for example, the rapid response to to a pandemic as is coming with vaccine being quickly made depend from science and the curiosity in observing the world and asking questions, was there, etc. Of course, this level, the level of science we do in this context needs a lot of training, what I do usually, but of course what we do is difficult to be understood in details from, let's say, lay people, while I stress the importance of starting very young with very basic scientific concepts. For example, what if you know the skin, cells, even DNA is concept that could be achieved in young children. I'm talking about biology mostly because this is my field. I'm not talking about math and physics. They are not my field, but they are equally important because constantly I do formulae, I use math to calculate my frequency, etc. And then we use physics law and to do our experiments, okay? So biology use math, physics and chemistry to grow, and we exchange everything. This is very important for our future and for our present, of course.
So here I am preparing a matrix that is used to view DNA. So from this I followed a protocol list. With this I will be doing a gel electrophoresis. So gel electrophoresis is whereby all the DNA bands that I've already extracted the DNA from the samples. In this case I've extracted breast cancer samples. So those samples, tomorrow when all of this is ready, I will be putting all those into the gel, and then that is where I will view all the DNA bands that are present, which is the specific DNA variations that I have talked about.
My name is Pajara Natro. I'm MPhil candidate in UP. I'm doing a project on the clinical pathological features of prostate cancer. I'm also working in several projects which includes HPV, cancer, and HIV. So this is our gel documentation system. After preparing our gel and running our DNA, so this is where we'll put our gel and then visualize it. So we are expecting to see the bands just like in this picture. So yeah.